Welcome to our seminar. My name is Erika Leonard and I'm the Director of Quality Control and Senior Research Histologist at Vector Laboratories, and this seminar will be considerations when designing your immunohistochemistry procedure. In the last section, we discussed how you select reagents based on the priorities you've determined for your laboratory. These priorities include sensitivity, simplicity, flexibility, rapid results, cost, and reproducibility. In this section, we're going to cover selecting ancillary reagents to help finish building your detection protocol. These ancillary reagents can include things like antigen unmasking solutions and primary antibody diluents, counterstains and mounting media, and blocking reagents for endogenous enzymes, avid and biotin, or nonspecific proteins. Well, when do I need to add these reagents? Well, certain reagents will be added based on the choices you've made for primary antibody and detection reagents. The manufacturers will provide information about ancillary reagents you may need to add to your protocol in order to achieve maximal and optimal staining. Other ancillary reagents you may identify as ones that could improve your results or improve your signal to noise. Other reagents may be added as you move through the optimization procedure required for this assay. The first ancillary reagent that we will discuss is antigen unmasking solution. Antigen retrieval ensures that your epitope is accessible to the primary antibody you've selected, and this is of primary importance in formalin-fixed paraffin-embedded tissue. There are several types of antigen retrieval that you may be able to do on your tissue section. The first is a protease-induced epitope retrieval, which cleaves peptide bonds at specific amino acid residues, unmasking the epitope, allowing your primary antibody to bind. A second type of retrieval is heat-induced epitope retrieval. This is thought to disrupt the cross-links and allow proper refolding of proteins, thereby allowing accessibility to your primary antibody. Common antigen unmasking solutions are citrate, pH 6.0, and tris, pH 9. As you can see in this example, on the left we get no staining using cyclin D1 with our detection system, but following a tris-based antigen unmasking step, we're able to see strong signal with our detection system on the right. Another ancillary reagent you may add into your protocol is a primary antibody diluent. These are typically protein-containing solutions which help to stabilize your primary antibody solution and can also serve to reduce nonspecific background of your secondary antibodies that you will apply for your detection reagents. Normal primary antibody diluents can include normal serum, animal-free blocker and diluent, or BSA. Another ancillary reagent you may choose to add to your detection system is a counterstain. Most cells are colorless and transparent, and counterstaining introduces a color to specific cellular structures to aid in the visualization and localization of your target. It also allows for better morphological visualization of certain cell structures. When choosing a counterstain, you have several choices, whether or not that counterstain is colorimetric or fluorescent. Also, counterstains can stain specific cellular structures. You want that counterstain to provide an excellent color contrast with the antibody immunostain of your detection reagents, and you want that counterstain to be compatible with the mounting media you have chosen for your immunohistochemistry assay. You need to optimize the staining intensity of the counterstain before adding it into your detection profile in order to make sure the staining is adequate. Oftentimes after an antigen retrieval step, the nuclei can be affected and reduce the uptake of the nuclear counterstain. There are several types of colorimetric counterstains available for you to choose. Hematoxylin, nuclear fast red, methyl green, and eosin are common choices. In this example, you can see that the hematoxylin counterstain, which is blue, provides excellent morphological detail when looking at the 5D3 staining in this human small bowel. There are also multiple fluorescent counterstains available for you to choose from. There's DAPI, propyridium iodide, and tritifolidin. In this example, you can see the blue nuclei of the DAPI staining and the red F-actin filaments of the tritifolidin give excellent morphological detail when visualizing the epithelial staining of this experiment. Next, you may need to select an appropriate mounting media for your immunohistochemistry assay. The mounting media in general serves to adhere a cover slip to your section, but it also protects the specimen from physical damage, from drying out, and may prevent photo bleaching when visualizing your fluorophore labeled detection reagents. There are several mounting media properties which you should consider before selecting a mounting media. The first is whether or not you need an aqueous or a non-aqueous based mounting media. This is generally determined by the enzymatic substrate or fluorophore that you've chosen in your detection reagent. Another decision is hardening versus non-hardening mounting media. This is also determined by multiple factors, such as the type and thickness of your specimen, your handling requirements, and storage requirements. A refractive index requirement should also be considered if you will be requiring a high-resolution visualization method. You also need to decide whether or not your protocol requires an anti-fade mounting media to protect your fluorophore from fading during visualization. Now we're going to talk about mounting media compatibility. You need the mounting media to be compatible with your chromogens and counterstains. This means that once you've mounted your sections, the color and intensity are stable over the duration of that storage period. The compatibility with fluorophores is also very important. You want a stable intensity of that fluorophore during the visualization of it, as well as during the storage. And you don't want the signal strength of your fluorophore to be affected by the mounting media. In this photo, you can see multiple antigens being detected fluorescently, counterstained with DAPI using our VectaShield hardset mounting media. There are several blocking reagents which also can be added into your procedure to optimize signal to noise. These are endogenous enzyme blocks, avid and biotin blocks, and protein blocks. And in the next section, we'll discuss these further as we optimize your procedure. In this section, we discussed choosing ancillary reagents to help complete your immunohistochemistry protocol. Unmasking solutions can make epitopes more accessible for binding. Blocking agents can stabilize the primary antibody and minimize nonspecific binding. Counterstains provide background color to aid visualization of the specific target staining. And mounting media help adhere cover slips to the slides and protect your specimen. In the next section, we'll discuss optimizing your protocol to maximize the performance of your detection system, as well as finalizing your ancillary reagent needs. Thank you for joining us for our seminar, and I invite you to listen to the additional sessions which are available on our website.